Here we are at part 2 of chapter 5. We were discussing the different types of photosynthesis, the different types of plants we have, and I wanted to go a little bit more in depth on that. So before we discussed that plants have stomata, these little holes where carbon dioxide can come in, so they can take the carbon dioxide, take that carbon part, and make sugars. There's like 6 carbon sugars. And we're looking at the differences a little bit. Most plants are C3, so what that means is the first thing that they convert the carbon and carbon dioxide into is a 3-carbon sugar. Uh, so they're under cool, moist conditions and normal light. They're the most efficient. They don't need lots of extra anatomy or enzymes. C4, the first thing they make carbon dioxide into is a 4-carbon sugar. This is a little more complicated, but they have more efficiency here, so they don't need their stomata open as much. They don't need as much carbon dioxide to come in. They photosynthesize faster because this carbon dioxide is delivered quicker into the 4-carbon sugar. So that's some plants like corn and a lot of our summer annuals. And then CAM photosynthesis has evolved in some very dry climate plants like cactuses, agave, orchids, and this is the most efficient water use. So during the day when it's really dry, their stomata are closed. They've got a waxy cuticle, they don't lose water. The stomata open only at night, so that's the only time they get carbon dioxide in. And so they lose less water, so they're able to be open in dry temperatures, and then the oxygen that they make is released at that time. And that's it for photosynthesis, chapter 7. Now let's look at enzymes. I've discussed enzymes a little bit, but we haven't actually really talked about what they do. Enzymes make reactions go faster. They define what our metabolism is. So metabolism is the reactions that occur in organisms. Very few reactions occur without enzymes. They go very, very slowly. So we need enzymes. They help make the reaction go faster. They're very specific. Each enzyme only helps with one reaction. So depending on what enzymes we have present in a cell, we can determine different types of metabolisms in different cells. So why do we need enzymes? Why do reactions go really slowly on their own? That's because they require activation energy to get started. So before we can do a chemical reaction, and remember that just means before we can make a bond, or sometimes even break a bond, we need to overcome the activation energy, this barrier. To make it happen, you have to put energy in. So what enzymes do is they decrease the activation energy. They make this upward slope smaller. So activation energy barrier, that's our reactants. Enzymes make it easier to go from one side to the other. And how do they do that? They have what's called an active site, where the substrate or the reactants bind in. You can see how they fit in there really nicely. So that's why each enzyme only works on one reaction, because it's got this active site, it brings them in, and then helps the reaction occur. So the example I like to do in class is I'll take 2 dry erase markers and I'll stick them together. And I'll say that we just built a molecule. And we have to think about how would this happen naturally? Well, first, those dry erase markers would have to come together in the right orientation. So even if they hit each other, but their ends didn't come together, they wouldn't form this bigger dry erase marker. They wouldn't form this new molecule. So what I did, I was like the enzyme. I took both the reactants and I put them together in the right orientation. So I decreased the activation energy. I was the enzyme. And notice that after the enzyme has done this, it looks the exact same as the beginning. There's no parts of the enzyme coming off. The enzyme can do this same reaction millions of times. And the word we use for making that reaction occur is called catalyzing. So here's a lovely view of that induced fit, the active site and the substrate, the reactants coming together, and they form this complex. And this shows how they can work over and over again. So enzymes available, we put the substrate, the reactants in, products are released, it can go again. And I like the analogy. An enzyme is like a wrench. That wrench is a very specific size for very specific bolts. And after you've tightened one bolt, you can go on and tighten the next bolt. You don't need to get a whole new wrench. It'll work over and over again. Okay, so review. Enzymes don't make things happen that wouldn't otherwise happen, but they make it go faster. They decrease the activation energy. And after they've done it once, they can do it again. And the same enzyme can actually do both the forward and reverse reaction, um, put bonds together or break them. But in biological systems, that generally doesn't happen. They usually only go forward. And that's because in our bodies, when we make one reaction, generally the product goes on to something else right away. So it doesn't sit there and go backwards. It keeps moving on. So there'll be another enzyme to take that and move it on. And then last, each enzyme is very specific. It only binds to its very specific substrate or reactant. And another good thing to note is that an enzyme's name usually tells you about what it does, and they generally end in the word part ase. So catalase, DNA unwindase, helicase, uh, polymerase, those are all different enzymes. They end in ase. So if you see that word, you can guess it's an enzyme that helps with a reaction. The next important part that we'll talk about is membrane transport. So we discussed before that the plasma membrane is selectively permeable, meaning that only certain things can cross. So that membrane controls what crosses. We'll have transport proteins in there to help allow some things that wouldn't normally be allowed to cross. That way we're able to regulate the internal environment of the cell, how much sodium is on the inside outside, getting sugar in to the cell, things of that nature. An important idea to understand when we're looking at the movement of molecules is how they want to move. So first we're going to look at passive transport. And what passive transport means is that we don't have to use ATP. We don't have to use energy to move the molecules. So in this case, diffusion is the first one we're going to look at. That's passive. Molecules move the way that they want to. We're not forcing them to do anything that they don't want to do. And this is going to be based on thermodynamics. So remember, molecules, they have heat, they're moving. They move around randomly. Remember entropy? So they want to spread out. And by moving around randomly, that's the best way to do it. So as they wander, they spread out. So you can see here, this is a great example of diffusion. When you put drops of food coloring, for example, into water, you can see that dye start to diffuse and move throughout the water until you've got all the molecules spread out. They're just kind of wandering around. So remember, it's passive. You don't need to put any energy in. So visually here we can see two examples of this diffusion. So you've got dye, and it's on one side of a membrane, and then you can see it spread from one side to the other. And then by the time it's equal on both sides, we've reached what we call equilibrium. So what that means is that at that point, there's not going to be any net movement either way. You're not going to have lots of red on this side and very few. It'll all still be randomly moving around, but for the most part, as we have a couple go this way, a couple will go that way, and it'll stay spread out. And this shows two types of molecules. So this purple is highly concentrated on the left half. It's got a very low concentration on the right half. So what it does is it moves down its concentration gradient from high to low, and it spreads out. to the right. Whereas the red dye has a high concentration on the right and a low concentration on the left, and it spreads down its concentration gradient from high to low to the left. And by the end at equilibrium, you've got everything spread out. So to reiterate that last point, molecules want to move down their concentration gradient. They want to spread out. When you spray perfume in a corner of a room, it's highly concentrated in that corner. And those molecules are going to diffuse down their concentration gradient, spread from where they're highly concentrated out to where they're lower concentrated. And if you've got students in a room, they'll go past the students and spread out. Another type of passive transport is facilitated diffusion. So this is for molecules that can't cross the plasma membrane on their own. So they want to diffuse and go down their concentration gradient, but they can't cross the membrane on their own. So they need some sort of transport protein, whether it's a channel, an open space that fits that molecule, or a carrier protein that grabs it from one side, changes shape, and sends it out the other. So they're still moving down their concentration gradient from high to low. They're still spreading out the way they want to move, so we don't need to put in any energy. It's still passive. And then the specialized case of osmosis. So osmosis is the diffusion of the solvent, generally water. Instead of diffusion like the solutes, the things that are dissolved in there, now we've got the solvent, water. So water spreads out. It wants to make the concentration of both sides equal. So if we put lots of solutes over here, lots of salt, for example, water would come this way to help make those equal. So here's a lovely example. We've got a little membrane here. We've got sugar molecules on each side, and so there's a lot more sugar on this side. So the sugar is highly concentrated. There's more space for water on the left, so the water has a higher concentration over here. It wants to make those two concentrations the same. But sugar can't fit through this membrane. It's too big. Only water can move through. So what you'll actually see happening is that water goes against gravity to spread and equal out those concentrations. When we compare concentrations of solutions, we use the word tonicity. So it's always a comparison. A hypertonic solution has a higher concentrate of solute. A hypotonic has a lower concentration of solute. So hyper, high, hyperactive, high energy. Hypo, low, hypoallergenic, low allergy causing. And if you're comparing two solutions and they have an equal concentration, they are considered isotonic. So here's our cell, and here's the solution. So in this case, there's lots more solutes in the cell, very few on the outside. So this solution would be hypotonic. Comparatively, the cell would be hypertonic. Whereas on this side, that's a hypertonic solution, high levels of solutes. Inside the cell, there's lower, so it'd be hypotonic. And here we've got isotonic, an equal amount of solutes inside and outside of the cell. And this is very important because we need to have isotonic solutions in our body, otherwise water's going to move to try to equal them. So for example, our red blood cells are really interesting. They are in our plasma, a watery solution, and when they're active, they've lost their nucleus. They have no way to regulate water movement. So our blood needs to be at the same level of solutes as the red blood cell. It needs to be isotonic. If not, water can enter or leave the cell and the cell can't control it. So if we put really watery solution here, that would be hypotonic. There's more solutes inside the cell. So water moves in to try to balance them out,